interesting aspects of being involved in UK Biobank, just seeing different people approach it in very different ways. So if I could ask the panel to come up. Bernard Keavney is going to chair it. He is the chair of the International Scientific Advisory Board for UK Biobank from University of Manchester. And William Astell, David Van Heel, Eric Meslin from the Ethics and Governance Council, where he's the deputy chair, and Nilesh Samani, to talk about genetics and beyond genetics. While you're just kind of settling down now, there were a few questions that maybe I could kind of answer. So we're already encouraging people to want to be in it. There's a question, are you going to open up the biobank to new participants? I'm 40 next year, smiley face. Unfortunately, no, but you could move to the US and become part of the Precision Medicine Initiative. And then there's a question about how do we enhance the data. So one question is around enhancing it by linking not to what you might think of as other medical or health-related records, but to other studies. So a number of participants in different studies and saying, well, can the data that has been obtained from me in another study be used to enhance the UK Biobank resource, or vice versa? And the answer is yes. We have already had some approaches from other studies that want to enhance their data by combining with UK Biobank in terms of the data that we have. And we are looking at opportunities to go the other way as well and get additional information from those other studies. And this is another area where there are discussions with the Ethics and Governance Council about how to manage that process. But it is certainly something that's being explored. And then another question related to enhancing the resource was around, well, what if I have a questionnaire that I would like to have administered to biobank participants? How do we go about getting that incorporated? Is there a sleep working group? The mechanism that we've got is that once the UK Biobank recruitment was going smoothly, we started to add in, as you saw, a number of enhancements. And these derived from work of the Enhancement Working Group, chaired by Paul Elliott from Imperial, with a number of people with expertise in different areas. They came up with a lot of suggestions, almost all of which we've now implemented or are implementing. And the Enhancement Working Group is now looking at, well, what next? What are the next series of assays that might be done on the samples? You heard from Naomi about infectious markers, but what about other markers that didn't get into that 40 or so set of analytes that are currently being run? What about environment? Again, can we link to environmental records? There was the question about meteorology. Can we find out more about environmental exposure? And there have been some discussions which we're now reactivating around whether there are ways of getting at exposures in individuals to various kinds of environmental aspects, be it pollution, be it radiation, and whatever. So if you have ideas, then please let us know, and we can push those through the Enhancement Working Group, get you to come along and help us to think about ways to enhance this resource. But I'll hand over to Bernard, and there is one question maybe, Bernard, that at some point during the panel discussion you could consider, and that is this question, how is impact being measured in UK Biobank, and at what time points? I know it doesn't just relate to the genetics, but perhaps this is one exemplar that you could touch on.
Thanks very much, Rory. One of, I think, the very important parts of today is not only to report back, for UK Biobank to report back to the participants and the scientific community on the progress that's been made, but also to encourage members of the scientific community to take up and utilise this great resource that so many people have put so much work into building. And I think you've seen throughout the course of today the fruits of that. And on the International Scientific Advisory Board, which I currently chair, we every year get an update from Rory and the team on how many applications have been made to use the resource. And something that has been very noticeable and very gratifying to see is that since the genetic data has been in prospect and then available, the numbers have rocketed up. This is clearly a resource which is of massive interest to the scientific community. So I hope that in our discussion in the next 20 minutes or so, there'll be additional points that will come out which will encourage members of the audience and the watching colleagues who may be considering using Biobank to get ahead and make an application to use the data. I'd like to start by inviting colleagues who haven't introduced themselves yet to do so.
Good afternoon. I'm Eric Meslin. I'm Vice Chair of the UK Biobank Ethics and Governance Council, and when I'm not doing that, I'm the President and CEO of the Council of Canadian Academies in Ottawa.
Thank you. I'm David Van Heel. I'm Professor of Genetics at Queen Mary University of London. We just made our first application to UK Biobank for genetic data. I'm also work at Barts Health NHS Trust, where I'm a consultant and clinical information officer for the trust.
I'm Nilesh Samani from the University of Leicester. I'm a cardiologist interested in genetics of heart diseases.
Hello, I'm William Astle. I work in William Owen's group at the University of Cambridge in the Department of Haematology. I'm working on two aspects of the genetic data. First of all, on a large GWAS, which combines UK Biobank data with data from Interval, which is a randomized controlled trial, which is run from our university. And also, we have an application in to try and sequence the top and tail, the top and bottom tails of the red blood cell count distribution. So we're going to whole genome sequence about 400 individuals from the two tails.
Okay, so we have some questions submitted. And the first one that I'd like to address is, do biobankers have access to their genotype data? Well, I think the answer is if you're a participant and you're wishing to have access to your individualized data sent back to you, I'm confident the answer to that is no. If you're a participant who's also a researcher, then perhaps as part of a project you might get your data anonymized with a lot of other people's data sent back to you. But perhaps I could use that as a kicking off point. Maybe, Eric, you could start to talk to us a little bit about the way genetic data has presented itself in the discussions in the Ethics and Governance Council, and perhaps the particular concerns surrounding that.
Sure, it's a great question, obviously not limited to UK Biobank. This is something that has been probably the focus of most biobanks around the world since their inception. The idea that one would wish to gain access to and make available this information is probably what we would call the holy grail of all of science. On the one hand, the greater the access and the more free one can get, then all of those nice SNP associations that we saw in the prior slide is where the action is. The more access to information, the more access to SNP data, the greater the likelihood of associations. Of course, the reason why it's such a nice challenge is that the more access, then obviously the greater the likelihood of identifying an individual, and by identifying them, raising all of the usual litany of ethics questions around privacy, confidentiality, and the like. What I think we find most intriguing and exciting about UK Biobank, and not just through the EGC, but I think most of us in this room, is that the individuals, many of whom who are in the room now, who have given of themselves literally pieces of themselves, have in a sense bound themselves by a kind of social contract to be participants in not just the physical data, but by hoping that it will be used for the greatest purpose. In fact, some of us were having a chat at the tea break before about whether the participants themselves are unique in a way that others who have not already agreed to participate might not be. So we've been having obviously very good discussions, and I hope useful ones for the UK Biobank around the way that individuals can present themselves, hope that their information will be used appropriately. We can talk again about consent later. But I think it's a fabulous opportunity to get clear on how access policies and how assumptions that the public might have about use and misuse can be laid to rest by really a very good protocol in place here at UK Biobank.
Thank you. Any other comments? David, would you like to? No? No? Okay. So I think that's a great point, Eric, that you make about the whole enterprise being founded on the altruism of the UK Biobank participants. Of course, there are some large impact rare genetic variants which have not been looked at in UK Biobank yet, which can increase risks of cancers and other serious diseases. Would any colleagues have any comments they'd like to make on how that might be handled?
So I think there's a lot of discussion in human genetics at the moment about feedback of genetic results to participants, and it's clear that some of the cancer risks that are found in studies of large multiply affected families might not apply to population, sporadic individuals in the population. And actually I think one of the things when we get primary care electronic health records in UK Biobank and combine that with the genetics, actually UK Biobank will be able to answer some of those questions as to what's the risk for sporadic cancer of this type or that type. And so actually, rather than feeding back results to participants, I think UK Biobank is going to determine the value of those results.
Mm-hmm. 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 One of the issues that came up earlier today was whether it might, under some circumstances, while preserving the necessary requirement not to feed back results, be able to enroll participants in studies that would select them based on genotypic information. So, Nilesh, would you have anything to comment on that?
I guess, I mean, I think that should be easy to do, relatively, especially for genetic variants where we are not sure about the precise relationship with disease, or whether it's one of many genetic signals for that particular disease. So if you partition people, that's fine. And you don't need to, I mean, we've had experience of this, you don't particularly need to tell them the genotype you're interested in. You say, you know, the way we normally phrase it would be to say, you know, we're interested in doing a study on blood cells or some other factor, and you may be somebody who we think would be suitable for such a study. In most cases, we get a very positive response to that. I think it's going to be more difficult coming back to this issue of very rare genetic variants, which may have much more an impact on disease. So if you're going to ask them to come back for very rare mutations, where you think the impact on the disease risk is quite high, then how you frame your invitation for them to come may be a bit more challenging. But I think coming back to the point you asked, Bernard, about transmitting information back to individuals who participated, particularly for rare things, you know, cancer risk. I guess it's the same issue that's come up with the MRI, the imaging. I don't see a differentiation between genetic information and the sort of findings. And I wasn't quite sure what UK Biobank's views were on the genetic information related to very rare conditions with high risk. So do we say that you're not going, you know, you seem to be doing it for MRI, or you might be doing it for certain MRI things, but you're not going to do it for any genetic tests, as far as I can tell.
Rory, would you like to comment on that?
Yeah, the approach that Biobank has taken in terms of feedback is, I suppose, to say that there are three different types. So during the baseline visit, participants were advised that measures that were being made where they could actually see the value anyway, they were told that those values would be provided to them at the end. So, for example, bone density measure, their respiratory function, their estimated body fat, and they were given those at the end. Also in the baseline visit, it was indicated to them that if there was an incidental finding during the visit that indicated a serious problem, which could be that the nurse might notice something that could be a melanoma, or noticed evidence of physical abuse, then that might be something that would be fed back in order to try to help with that incidental finding. But the information material and the consent was very clear that subsequent to that, there would be no feedback of any of the findings from the use of the data. So that's been the approach that was also adopted in the imaging visit. The standard measures that are being made, that are being repeated from the baseline visit, are being provided, slightly tweaked because there's some slightly different measures. The incidental findings becomes a much bigger issue because although in the baseline visit it was part of the process, there were relatively few. But of course with imaging, as we saw, depending on how you approach them, those incidental findings could be very large or not. But the approach that Biobank took was that they should be incidental and that they would be things that would be noticed by the radiographer during the process, confirmed by a radiologist, and then reported. And that was the protocol that fitted with the original process. We felt that we should test that to see what was the balance between net harm, net benefit, as Jonathan Sellers mentioned, against approaches that have been used in other imaging studies, for example, in the German national cohort, where they're doing systematic screening by radiologists of all the scans, and for the reasons that were presented, came to the conclusion that the incidental finding approach was probably an appropriate one, not necessarily the only one, but an appropriate one on this scale. But that still subsequent analysis of the images, as with the genotyping, as with the biomarkers, would not be fed back. That's not to say that these things can't be revisited, and this is an area that is discussed with the Ethics and Governance Council. But there is a feeling that participants gave consent on the basis that there would not be feedback, so that if there was a change in that, Eric may well want to comment on this, then there would be a need to change the discussion with participants around consent. Sorry, long answer too, but a very complicated area.
Eric, any further comment?
Just to agree with Rory's point that today's incidental finding is tomorrow's more understandable finding, and I think the suggestion that we have a protocol in place that was designed for a particular purpose, designed to both protect the rights and welfare of those who are participating and obviously promote the best interests of science. Of course that can be revisited. That's why you have a fluid system where there's interaction on a regular basis between the EGC and the PI and the biobank. I think Rory's point is spot on.
So Jonathan, I think we have a couple of questions which will be particularly directed to you. A colleague has asked which reference panel will the full 500K data be imputed to? And for those who already have access, will it be a relatively quick and simple process to update the current data set to the 500K?
Well, okay, so the first question, the first release of the genetic data, we used the UK 10K reference panel, which was, well, a combination of that reference panel with the 1,000 genomes reference panel. And that was a kind of pragmatic decision at the time. It was the best available reference panel we had. It combined SNP genotypes with short indels. So researchers have been able to test both SNPs and short indels when looking for associations. But in the subsequent year, the Haplotype Reference Consortium has kind of come online, and this is a much bigger reference panel. So it has many more individuals in it. It's a kind of deeper reference panel, but it has a rather smaller set of SNPs that we're more confident in that we can impute. So the current strategy is that we'll use the Haplotype Reference Consortium panel for the SNPs in that panel, and then for all of the SNPs and indels not in that panel, we'll impute from the combined UK 10K and 1,000 genomes resource. So we're doing the best we can to get the best of both worlds. So it will be a larger set than currently is available, but all of the SNPs and the indels that were in the first release will be in the subsequent release. And what was the second question?
The second question was, would it be a quick and easy process to update the current data set?
We hope so. We're working hard to provide the data in a format that links in with all the analysis tools that people use. We're doing work with colleagues in Oxford to try and make that quicker. When the data set becomes online, there will be, it's quite a large amount of data in terms of terabytes of data will need to be uploaded. But we don't foresee any large problems at all. It should be seamless.
And another question about the SNP data has been asked, which is some SNPs were selected for certain disorders, but at least on the UK Biobank resource, there isn't an indication of which they were. Is there any guidance that is there regarding which SNPs went on the chip for what reason that colleagues can access?
I think there is quite a detailed document that details the design process that you can obtain.
You would refer the colleagues to that?
Yes.
That document. Okay, that's great. Well, maybe it's a good point now to refer to to address the question Rory asked about what is the likely impact of UK Biobank. How should we measure it, and when should we expect it to be evident? Clearly, improving the health of future generations is I'll give you a little bit more time to think about that. Is the tagline for Biobank, and we hope that the impact will be not in the far too distant future. I think we've seen some very interesting in the talks we've had so far, actually, evidence of potentially important impacts. Jess's talk certainly went beyond genetics in terms of looking at potential social policy issues, I felt, also, Martin's presentation highlighted that several novel drug targets have been potentially identified as a result of the UK Believe study. And perhaps another thing which we might discuss is the potential utility of multi-SNP risk scores for risk stratification in common disease. So would anybody like to take up any of those areas?
Well, this is a simple answer, of course, is the impact is not going to be measured by one thing. I mean, I think UK Biobank's already had an impact in the way we do research in this country, the way the world thinks about doing research in terms of scale. So there's already impact there. But I guess it's going to take, I mean, how we should show you an impact already. So there's a new drug, some of you will know, that's come on the market for lowering your cholesterol, which people who can't take statins can use, called a PCSK9 inhibitor. And the worry, of course, is will this drug have side effects? And I can tell you, using the genetic data that UK Biobank have collected, you can tell, looking whether a variant that affects this gene has effects on other diseases. And you see a very clean signal, suggesting that this drug is going to be safe, or likely to be safe in the longer term. So there's lots of impacts. There are lots of impacts that UK Biobank will have in different ways, and I don't think you'll be able to collate. I don't think Rory and his team are going to be able to collate all of them in a single way. The impact will be different for different communities. For industry, there'll be different sorts of impact. For the public, it'll be different. For people who work in public health, it will be different. So I think the question is well intentioned, but it's going to be very difficult to judge impact in any simple way.
Yes, I think some of the impacts might almost be things that didn't happen. It's always difficult to measure something that didn't happen. Yeah. For example, the potential that the genetic data gives drug developers to carry out Mendelian randomization studies and sort causative risk factors from those that they really shouldn't be bothering targeting with drugs. And I guess we've got some good examples of that already. David?
I think you're going to get a whole new area of impact when you start to get the primary healthcare data from the GPs, for example, combining that with genetics and some of the blood measurements for things like drug response, adverse drug reactions, and sorry to say it, but as the UK Biobank participants age, they will be exposed to more and more medications, and the data on what can be discovered will increase.
Yes, and actually that was the final question that we had from the floor, which was how much will we find by combining genetics with primary care e-health records? So you've got ahead there. Another aspect of the sort of beyond SNPs, beyond genetics, are other things that can be measured in DNA. Nilesh, you've been working on telomeres, and I wondered whether you'd maybe like to tell us about that and present it perhaps as an example of how the way in which you've worked with UK Biobank to enhance that, working from an independent research group perspective.
So, well, I hope there's an opportunity in the next meeting to present some of the early findings with the telomeres. So those of you who don't know what telomeres are, these are the ends of your chromosomes, and we all vary in the length of our telomeres. And there's some circumstantial evidence suggesting that your length of telomeres determines your risk of getting chronic diseases. It's a marker of biological aging. So, as you heard earlier today, there may be 3 people who are 50, but they all look different. And one way you could be is you could be biologically, or in terms of your age, be different. So we've had the opportunity and the privilege of measuring telomere length in all 500,000 subjects in UK Biobank. And this is an ongoing project. We've done the first 100,000 so far, and hope that we'll complete the rest in the next couple of years. And really the opportunity just shows there are opportunities because Rory and his team and Tim Peakman plated out the DNAs, extract the DNA from all 500,000 people and send them to Affymetrix to have the genotypes done. And if I have some sort of eureka moments, there are 1 or 2 that happen in the shower, just thinking about how we could use the DNA that was sitting at Affymetrix and we're going to be put in the dustbin after the genotypes have been achieved. And of course, each of those DNAs took a pound or 2 to put on a plate. And so I said, well, we can measure telomere length in these individuals. And we were able to get all the DNAs back from Affymetrix, and do this. And at a much more cheaper cost than it would have otherwise. So we think that one of the interesting things, of course, with our aging population is, do we all have different trajectories in the way we biologically age, rather than our chronological age? And telomere length may be a marker of that age, biological age relationship with chronic diseases.
Will. So I was just going back to the impact of UK Biobank. I think one of the main advantages of the study from the genetic point of view compared to previous designs is the access that we have to the individual level data. So particularly for the quantitative trait analysis that we've been doing with Nicole on blood traits, Nicole Soranzo, because we've had access to the individual level data, we've been able to model out a huge amount of technical variation, which has given us a lot more power. And because the sample size is so large, we've started to be able to get down to detect rare variants. Frequencies of 1 in 1,000. And the effect sizes there are very large, so they're biologically interpretable. Rare variants, it tends to be much easier with a rare variant to identify the causal allele, to understand the LD structure, and therefore to understand the biology and follow-up experiments.
So that leads us, I think, into the sort of the next wave, perhaps, of genetic analyses, potentially at UK Biobank, whole genome sequencing. Where should we be going in respect of that?
So I had a technical question for UK Biobank, actually. I'm interested to hear William has got a sequencing application into UK Biobank. I've got an exome sequencing application into UK Biobank. I suspect others will have as well. In fact, I suspect our sequencing is being done at the same place. How do you ensure data quality? And then also, exome or whole genome sequencing is very expensive at the moment. How can you save money for us and the research councils by not sequencing the same subject 10 times for different studies?
So I think the example of UK Believe is a nice one. So that was a proposal from a research group to do genotyping 50,000 participants. It really stimulated the International Scientific Advisory Board and through them the funders to recognize the value of actually turning samples into data, because data are much easier to share than our samples. And I think that strategy also then led on to them funding the first set of biomarkers, HbA1c, the urine markers, some biomarkers in the serum. And it's why we are thinking about, well, how do we do that for another set of markers, the infectious markers that Naomi Allen mentioned, possibly also another set of kind of standard biomarkers around, say, BNP and various other measures of nutrition. But I think the same issue applies with respect to proteomics, metabolomics, the epigenome, and sequencing. So I guess our job in Biobank is to try to coordinate those activities to avoid using up depletable sample, but wherever possible, when the research community is saying this is now the time to act, to use the International Science Advisory Board and to work with the funders to say, well, now is the time to act, rather than doing this in subsets. Is there a way of a consortium of government, charity, perhaps now commercial funders, to work together to do something on the whole cohort, such that the data can be used for all kinds of different researchers. And again, this illustrates the way in which UK Biobank is an experiment in so many different ways. How do we keep building this resource, driven by the research community, driven by the opportunities, but coordinate it so that funding is used as effectively as possible. And we've been fantastically helped by the Wellcome Trust and the MRC helping to coordinate that process. But I think now at our 10-year anniversary is a real opportunity to bring more funders, and as I say, beyond government and charity, more funders into this to turn sample or imaging data. We've seen examples of industry getting involved in turning imaging data into accessible information. So we look at the applications and we try to tell people, well, there are other groups doing that. Do you want to be put in touch with each other? But we're also telling the funders and telling the ISAB, this is the way in which things are going. And also we're looking to the research community to say, what should UK Biobank be doing? I mean, it shouldn't be a passive process. It should be the research community saying, look, this is now the time to act.
I think you should be exome sequencing the first 100,000.
So just in closing, I'd like to invite comment from the floor from any participants who are in the audience still, who might like to say anything, not be a question, a comment would be very welcome about anything that they've heard about the genetics side of UK Biobank today. Have a hand up there.
Willem Ouwehand, Cambridge. Just back to the imputation. Will and I have been discussing whether we should impute against the first 10,000 whole genome sequence samples of the 100,000 Genomes project. So I've been talking with the Oxford team about this. We're testing the algorithms at the moment. Or they are testing the algorithms so that they can cope with the dense data. But don't we think that if we want an ideal reference set, that the 100,000 Genomes project is a more stronger driver for accurate imputation, particularly in the non-coding space, and particularly in gene desets where you don't have that many, you completely rely on your scaffold to impute from?
Yes, so the 100,000...
Just speak, I think.
Okay, the 100,000 Genomes project data isn't available yet, so...
It is. I've given access to Oxford, so for the first 10,000. And Jonathan Marchini is working on it.
Yeah, that's me.
Oh, sorry.
So, yeah.
Yeah, so you have, I think there is access, right?
Well, it hasn't, we haven't phased that yet, Willem, so, yeah.
But it doesn't take away the debate whether we should, I was talking with Mark Caulfield, we are probably on 20,000 genomes by August, whether we as a community should say, if we now go to impute again, that we should impute against that reference set, or do we actually say what your line, your approach was a completely different one?
Well, we have to make a pragmatic approach in the time we have, the time scale we have. What's the available reference panel now for imputation? Has it been tested? These things take time. And right now we think that the HRC panel that has been used throughout the community throughout the last year or so is a very stable and good approach. There's nothing stopping us in the future from re-imputing Biobank from a future panel. But I think given the kind of agreed timelines that are in place for the data release. Right now, the HRC and UK 10K plus 1,000 genomes reference panel is the best approach to take.
Yeah, although on rare variants, I think we need then to be very careful.
Okay, thank you. I think in the interest of time, do we have a comment or a question from a participant? If not, I'll close the discussion there. Thank you all very much.
You'll be pleased to know I'm not going to try and round up such a wide-ranging discussion during the morning or the afternoon, but just to thank everybody for participating in this first meeting. We hope now that as a 10-year-old, UK Biobank is mature enough to have annual meetings. It is now a prospective cohort. I think that's the critical thing. So in terms of the question about impact, which is an important question, it's easy to assess impact in terms of numbers of publications or numbers of abstracts presented. But really, what we're trying to get at is impact on health. And what UK Biobank was established for, why the MRC and the Wellcome Trust have put so much behind it, and so many people have put so much time into it, is in order to develop a prospective cohort that allows us to look at the determinants of disease in the long term. And now, as we start to get linkage into this wide range of health records, as we get to the point where we anticipate being able to link over the next year to the primary care records as well as the death, cancer, and hospitalization data, then I think we will not have to ask the question about how you determine impact, because it will be self-evident. So I look forward to having another of these meetings in a year from now. We would very much welcome your feedback on how this meeting was set up, whether it works, what would be better, how it might be different. But I'd like to just thank you very much for your participation in it and for all of your participation in UK Biobank. Thank you.